Hi, my name is Cecilia Reed, and I'm a student in Survey of Biochemistry this summer, and my hot topic presentation is semaglutide. First, our agenda, I'll talk about why I chose semaglutide, then move on to the structure, and then talk about how it interacts with our metabolic processes, and then I'll wrap up. So, charging right ahead, why semaglutide? Semaglutide has become a major hot topic in the world of biochemistry. It was approved as a drug to help manage type 2 diabetes in addition to both diet and exercise management, but has since gained popularity as a weight loss drug. It was first approved for injection in December of 2017 and then was approved for oral ingestion in 2019. Semaglutide, which is better known by its name brands, Wegovy and Ozempic, is the first weight loss drug approved since Saxenda in 2014. Importantly, it has only been approved as an adjunct to diet and exercise, but has exploded in popularity due to the rise of celebrity use and weight management. But what does semaglutide actually do in the body? How is it so effective in helping people manage their weight and type 2 diabetes? In order to answer this, we'll need to take a look at semaglutide's structure. Semaglutide is a polypeptide chain, which belongs to proteins in the building blocks of life. Proteins are normally described in terms of their 2D structure, or alpha helices and beta sheets. Their 3D structure, such as alpha and beta proteins, or a mixture of both, and their 4D structure, such as homotrimers, heterodimers, heterotetramers, or dimers of trimers, and etc. Semaglutide appears to have a mixture of beta sheets and alpha helices, as well as random coils connecting the two. According to the rendering of the 3D structure from the Protein Data Bank, the tertiary structure appears to be a mixture of alpha and beta proteins. Since semaglutide is a polypeptide chain, it is not described as having a quaternary structure. Its polypeptide backbone is comprised of a 31 amino acid chain with special modifications that differentiate it from the naturally occurring GLP-1 hormone. The alanine at position 8 and the lysine at position 34 are replaced with amino isobutyric acid and arginine. It contains a C18 fatty acid side chain, which helps semaglutide bond to the plasma albumin, prolonging its half-life in the body, making it suitable for weekly injections. The C18 acid side chain is hydrophobic, but the histidine at the end of the molecule is capable of forming hydrogen bonding. This hydrogen bonding makes it capable of being partially soluble in water, which makes it an amphipathic molecule. Next, let's look at how semaglutide interacts with our metabolic processes. Semaglutide is a GLP agonist, meaning that it mimics glucagon-like peptide-1, a hormone that is produced by the small intestine. GLP-1 is released around 15 to 30 minutes after you start eating. Its main purpose is to stimulate insulin production and block glucagon secretion. Let's break this down. Insulin is a hormone that regulates gluconeogenesis and blood sugar levels. It is released by the pancreas to prevent blood sugar levels from getting too high. It accomplishes this by suppressing gluconeogenesis, which pumps glucose back into the bloodstream. So what is glucagon? Glucagon increases blood sugar levels by signaling to the liver to convert glycogen into glucose by promoting gluconeogenesis, which the body can use for energy. It works together with insulin to regulate blood sugar levels and glucose levels. Here's a bird's eye view of gluconeogenesis, which converts pyruvate to glucose back to glucose in 4 reactions. Back to GLP-1, which suppresses gluconeogenesis and triggers the production of insulin. Insulin stores glucose for energy and prevents blood sugar spikes after eating, which are associated with weight gain over time. It may be confusing initially why preventing the conversion of stored glucose would assist in weight loss, but it is actually GLP's second effect that is so important for weight loss. A secondary effect of GLP-1 is that it binds to hormone receptors in the brain that regulate hunger. Regulate hunger. GLP-1 effectively makes you feel more full, causing you to eat less and results in a caloric deficit. This deficit is actually what causes weight loss associated with semaglutide. GLP-1 also slows gastric emptying, which can help reinforce the feelings of being full, of fullness and satiety. And it also slows the absorption of glucose into the bloodstream, also assisting in weight loss. Now let's wrap this up. Semaglutide is effective in mimicking GLP-1 polypeptide, which is effectively able to trick the body into believing that it is full by binding to receptors in the brain and boosting insulin levels. It also slows gastric emptying, which again contributes to the feeling of being full, and also prevents spikes in blood sugar, which is associated with weight gain over time. The widespread success and popularity of semaglutide is palpable, with around 1 in 8 adults in the US having been on it at some point. Semaglutide is certainly a promising medication as an effective way to manage weight, but research is still being conducted into the other effects of the drug. Some patients on semaglutide have reported that they feel less of a compulsion to gamble, drink alcohol, smoke or consume nicotine, and even online shop. While semaglutide is not approved as a method to manage addiction, it opens an interesting avenue for future research into addiction management. As semaglutide continues to increase in popularity, it will be interesting to see what other effects it will have on people's behavior and what doors it could open in exploring how our metabolic processes influence our behavior. Thank you very much, and here are my works cited for the pictures and the sources, which I will put in the YouTube video description. Thank you so much.